Hi, everyone. We're in section 3.3, and we're going to talk about the plasma membrane and how substances cross it. The first thing we're going to talk about is to describe the structure of the plasma membrane and list the types of molecules found in the membrane. And this little introduction right here is part of what we already looked at in an earlier section of this chapter. But it says, like all cells, a human cell is surrounded by a plasma membrane. The membrane's a boundary between the inside and the outside. And when we were talking about organic molecules, we were saying that lipids are not soluble in water. So if you have a cell that's surrounded by a solution containing water in it, and it has solutions containing water outside of it and inside of the cell, it makes sense that most of the cell membrane would be, or the outside boundary of the cell would be made of lipid. So you've probably heard of these molecules called phospholipids, and all living organisms have them. And they have a cell membrane or a plasma membrane with 2 layers of phospholipids. And there's a lot more details about that with hydrophobic and hydrophilic areas in the phospholipids, but I'm not going to go into that for our discussion. I just want you to know that there's 2 layers of phospholipids making up membranes. There also are these big molecules or proteins, and proteins can do all sorts of things. They can be receptors, they can be transporters. And the third and final major molecule found in plasma membranes is cholesterol. And cholesterol, they have them drawn here as these little flat purple molecules. And we always think of cholesterol being bad stuff, but it turns out that cholesterol is actually important in our diet, and it helps stabilize the plasma membrane. I like to think of the cholesterol in the membrane similar to, I know they used to have water beds. They're not as popular now as they used to be, but you jump in a water bed and all the water kind of sloshes around. Well, somebody got this great idea that you build kind of vertical sections in the water bed to keep the water from sloshing around. And cholesterol is kind of like those vertical sections you would put in a water bed. It kind of stops the plasma membrane from moving around a lot, and it stabilizes it. So it's important to have cholesterol in our diet. The unfortunate thing is most of us eat too much of it. And so there's a little discussion about that. It also mentions that model that I just described is called the fluid mosaic model. The main reason why I wanted to talk about the membrane is to talk a little bit about how molecules move into and out of the membrane. And there's a lot more detail here, and I just wanted to touch on 2 major things called diffusion and osmosis. Now, you're familiar with the concept of diffusion, whether you realize it or not. It's just the random movement of molecules from a high concentration to a low concentration until they're equally distributed. And you're familiar with the idea of, oh, somebody is wearing perfume and sits down in the classroom, and you don't smell that perfume right away, but gradually just the people close to that person will smell it. And then the people that are a little bit farther away and a little bit farther away over time. And so that perfume's moving from high concentration from the person's body to lower concentration diffusing through the room. This video is a good video to watch talking about diffusion, and it goes into something called facilitated diffusion, which I'm not going to be talking about, but the beginning part about diffusion is helpful, and I would recommend reading this section. The diagram here is just trying to show you that diffusion of molecules, here's the plasma membrane, can happen through a plasma membrane of a cell. So you can see here the concentration of molecules is lower, and over time you're going to get an equal distribution of molecules on both sides of the plasma membrane. I kind of neglected saying the overall function of the plasma membrane really is to just control what goes in and out of the cell. Without a plasma membrane, the inside and the outside of the cell would look identical. Okay. So the next thing I want to talk about is, here's another little video talking about diffusion you can watch. The second thing I wanted to talk about related to plasma membranes is osmosis. Now, we did a section of osmosis in the lab book, And I tried to, there's some things to watch, there's some virtual labs, and I did a little discussion about the movement of water and some pictures of how water and moves in and out of the cells. I think that's how I would normally explain it to a class. So definitely, if osmosis, if you're just starting to understand osmosis and it's not completely clear, definitely go to the lab materials and look at those. That can help you understand osmosis also. Osmosis is a really important concept for health. It explains a lot of things, health situations that you might be familiar with, like why somebody's feet might swell up when they eat a lot of salt or something like that, or when you retain water when you eat a lot of salt. Um, and it also is really important as far as the function of the body. If you're going into nursing, you will learn about osmosis, and you will talk about it in microbiology. You'll talk about it in your nursing class. So it's a good one to understand. The definition of osmosis is the movement of water across a selectively permeable membrane. So let me just go back. Osmosis is the movement of water. When we are talking about diffusion, many different kinds of molecules can diffuse, but only water moves by osmosis. The other one, here they have the net movement. You can use the word diffusion there. So osmosis is the diffusion of water, and it's across a selectively permeable membrane. That means a membrane that only allows some things to go through. So a plasma membrane is exactly that, a selectively permeable membrane. And it says the direction of water will diffuse. The direction the water will diffuse is determined about the tonicity of the solutions. That means the concentration of the solutions. So if you have a solution that has a high amount of salt, it's going to have a relatively low amount of water. And diffusion is always going to move molecules from high concentration to low. So in osmosis, water is going to move from a solution of low water concentration to higher water concentration. Well, how do you know if the concentration of the water, if you have a high amount of salt, you have a low amount of water, or if you have a high amount of protein, any kind of solid, well, many kinds of solids, solutes, but if you have a high amount of protein, you have a low amount of water. There is a video here that talks about it, and like I said, there's some information in the lab that also will help you. But what you're shooting for is trying to understand what's going on in this diagram here, this picture. And there's three different kinds of solutions. There's isotonic, hypotonic, and hypertonic. An isotonic solution is one where the concentration of water is the same inside the cells as outside. Iso, same. So our body fluids are isotonic to our cells. So diffusion says in diffusion, water molecules will move from high concentration to low. Well, the concentration's equal here, so every molecule of water that moves out of a cell, another one's going to move in. And the cells are going to look normal and happy. And this is a normal looking red blood cell. There's a white blood cell right there. If I put red blood cells in a hypotonic solution, it says lower solute concentration. So, for example, I could take red blood cells and put them in pure water. And hypotonic means there's a lower concentration of solute, there's a lower concentration of salt outside of the cell than inside of the cell. Now remember I said if there's a low concentration of salt, there's a high concentration of water. Osmosis says water moves from high concentration to low, so water will move into cells when there's a hypotonic solution. And what's going to happen? Those cells are going to swell up, and they actually could even burst. And that is why when somebody's dehydrated, you don't give them an IV containing 100% water, because you can cause their cells to pop. Too much water is going to move into the cells too fast and lyse or break open the red blood cells. If I take red blood cells and I put them in a hypertonic solution, hyper sounds like high, and that means a high salt solution outside of the cell or a high sugar solution. If I have a high amount of solute outside of the cell, that means I have a low amount of water. So the water inside the cell is at a higher concentration, and osmosis says water moves from high concentration to low. So my cells are going to shrivel up, and there's a word called crenate or plasmolyze. You hear both of those words, but you can just say shrivel up if you want. And these cells are going to shrivel up, and you can imagine that that's not a good thing. Again, if you get, somebody had too much salt, it could cause cells to shrivel up. Now, what our bodies do is our kidneys regulate the amount of salt and water in our body fluids, keeping our fluid or doing what it can to keep our fluid at an isotonic solution. So there's again another little video, some pictures, and more information on different kinds of transport, which we are not going to do. But there's some questions at the end that you can look at. Mostly there's just this first one. And make sure you go to lab and look that over too. Good luck.